We worked in many projects together.
Oh, yeah.
Yeah.
I can see like you've done everything related to chickpea, almost. You're a pioneer in that. Yeah.
Actually,
Everybody have joined Chaitanya?
Yeah, ma'am. People are getting joined. So hope we can start the meeting. Right.
I
Abhigna?
think we have 5 more minutes to go.
5 more minutes to go.
Yeah, because we are broadcasting from YouTube, right? From Zoom to YouTube,
Yeah,
right? So it will take
yeah.
the lag.
Does it? Yeah. Yeah.
Abhigna, could you please switch on your cam? Yeah.
Am I visible?
Yeah, you are visible.
Okay.
Yeah, we will wait for 2 more minutes otherwise.
Yeah, that will be fine. There's some problem with the Wi-Fi here. I don't know what's going on.
We are on live actually present. So we shall be starting in
sorry.
1 or 2 minutes. Yes, Abhigna, I hope we can start now. Please go ahead.
Good morning, everyone. Distinguished participants, ladies and gentlemen. I'm pleased to welcome you to the webinar on Advances in Chickpea Improvement and Impacts of Improved Varieties on Chickpea Production. In this virtual conference room,
Yeah, you can go.
am I audible now?
Yeah, you're audible.
Shall we start?
Yeah, already we have started. Now we are on live. You can continue with that.
In this virtual conference room, I see such amazing guests and dignitaries from around the world, sharing same interests and enlightening one another with the advancing in the field of agriculture. I'm sure we are also reaching out to many new practitioners, technologists, innovators, business professionals, and civil society representatives, fellow mates, and many more people. We all understand the importance of science, technology, and innovation in our day-to-day lives and the ways in which they're transforming the world. If this was not clear earlier, it is blindingly obvious now, alongside the intricacies arising with the hour at hand. Science, technology, and innovation are indispensable for a response to and upliftment from the current and forthcoming scenarios. Equally important are deployment of scientific inventions, innovations, and technological solutions on a scale that will reach their target audience. Why here? Why today? What is the purpose? We are all embraced by the presence of distinguished and well-rounded dignitaries to shed some light upon the varied measures to be taken under consideration for the distinct genomes and the selection of crops based on the same. All our online webinars aim to discuss leading-edge technologies and recent scientific developments in addition to the immediate challenges in agriculture and elite sectors. The idea is to familiarize students and professionals about current research trends, relevant and pioneering technologies in plant genomics for high-level analysis and interdisciplinary areas that are embraced in leading laboratories by the excelling scientists. Plantgenomia. It is a non-profit organization promoting and publicizing plant sciences and investigations in the field of agriculture across the globe. We organize various webinars and developments comprehending plant research for all the students, researchers, industrialists, and professors internationally. Here at Plantgenomia, we hold a global team of reputed scientists and research scholars from various parts of the world, distinct states of India, the United States of America, United Kingdom, Nigeria, Philippines, and Spain, etc. Technical support is laid to us by the scholars from the Department of Genetics and Plant Breeding. Our motto is to disseminate every bit of knowledge among all. We believe that no research is accomplished until broadcasted, and that it is an ongoing process through layers and lapses of time. I would also like to extend my heartiest welcome to the senior dignitary, Professor Pooran Gaur, Adjunct Professor, the UWA Institute of Agriculture, University of Western Australia, and former Director, Research Program Asia at International Crop Research Institute for Semi-Arid Tropics, ICRISAT. The core subject specialist team, the core support team, our esteemed founder, Mr. Alamoru Krishna Chaitanya, and the lovely audience. Now we have the much admired founder of Plantgenomia, Mr. Alamoru Krishna Chaitanya, to address the audience and guests present with us today and to remark the initiative taken up by Plantgenomia and his team. Over to Chaitanya.
Thanks, Abhigna. I hope I'm audible and clear to all.
Yes, you are.
Fine. Good morning, good afternoon, and good evening to everyone who are watching us here in this virtual gathering all over the world. Welcome you all once again on behalf of complete Plantgenomia and team. And I am delighted to say you that Plantgenomia received a good number of registrations from almost 30 plus countries across the globe. With an aim to promote plant science research, Plantgenomia is organizing the talks from international renowned scientists who are well known for their research skills. Also, very soon we are about to launch our scientific forum for getting the guidance from scientists to the selected participants in writing scientific manuscripts, papers, books, chapters, proposals, and many more in various disciplines of agriculture. Plantgenomia is all about knowledge dissemination and would be a package of library of several such initiatives. We thank you all for the kind support and participation. I must truly thank all the scientists and speakers for the guidance and encouragement to do many such activities. Today, we had a well-renowned speaker who is internationally recognized scientist and an efficient plant breeder going to deliver a talk on advances in chickpea improvement and impacts of improved varieties on chickpea production. As we all know that we are day by day overcoming the food security, but still staggering with the nutritional security. This topic is very much needed and to be discussed in this regard of global climate change scenario. I heartfully thank Professor Pooran Gaur sir for accepting our kind invitation in very less time out of his very busy schedule. Thank you.
Thank you very much, Mr. Alamoru. Those were some very gripping words coming from you. While we are presenting a gesture of gratitude, I would also like to thank Dr. K.P. Vishwanathan, Vice Chancellor of Mahatma Phule Krishi Vidyapeeth, Rauri, and Dr. K.V. Peter, former Vice Chancellor of Kerala Agriculture University, for giving the valuable forward message on Plantgenomia webinar. Before we begin, I would like to introduce Dr. Rupali, who had graduated with Ph.D. from the University of Western Australia and presently working at the Department of Agriculture and Food, Western Australia, to introduce our revered speaker, Professor Pooran Gaur. Over to Rupali, ma'am. Ma'am, you're on mute.
Can you hear me now?
Yes, ma'am.
Yes, ma'am.
Hello? Yeah.
Yes, ma'am.
Okay, thank you, Abhigna. On behalf of Plantgenomia, it is my greatest pleasure to introduce our speaker, Professor Pooran Gaur. Professor Pooran Gaur is a former director for Research Program Asia under ICRISAT, which is International Crops Research Institute for the Semi-Arid Tropics, India. He is currently an adjunct professor at the University of Western Australia Institute of Agriculture. He obtained his Ph.D. degree in crop science from University of Saskatchewan, Canada, where he developed the first genetic map of chickpea. Professor Gaur joined ICRISAT in 2001 as a chickpea breeder and geneticist And before joining ICRISAT, he was an associate professor at Jawaharlal Nehru Krishi Vishwavidyalaya, Jabalpur, India. His entry into chickpea research was at the time when the crop was commonly referred to as an orphan crop. Dr. Gaur is a famous chickpea breeder and has over 3 decades of experience in chickpea breeding and genetics. And he has been instrumental in releasing 68 chickpea varieties from ICRISAT bred material. Professor Gaur work on extra large seeded Kabuli chickpea has been helpful to meet domestic demand in India. He has been a pioneer in chickpea research and has released 3 machine harvestable varieties for the first time in India. These varieties have helped reduce the cost and drudgery in chickpea farming, making it attractive for farmers. Additionally, his team was instrumental in developing heat-tolerant chickpea varieties. And to sum up, his research on chickpea in India not just showed up production, but also improved exports of Kabuli chickpea. Apart from his work in India, Professor Gaur and his team have also helped increase Myanmar's chickpea production by releasing early maturing varieties. For his overall impactful work on research and leadership in chickpea breeding, Professor Pooran Gaur has been awarded the Durian Margaret Mashler Award, which is ICRISAT's highest scientific honor in 2019. Dr. Professor Pooran Gaur, thank you for accepting our request to be the distinguished speaker for today's webinar on advances in chickpea improvement and impacts of improved varieties on chickpea production. It is our extreme pleasure to have you today, sir. And I now request you to share the screen to our audiences and proceed with the talk. Thank you, Professor, and over to you.
Thanks, Rupali. And good morning, good afternoon to everyone. Good evening, wherever you are, depending on your locations. My appreciation, high appreciation to the Plantgenomia team. I hope you can see my screen now.
Yes, sir.
Yes.
Okay. Yeah. So I must compliment the young team of Plantgenomia for launching this platform. My appreciation to Alamuru, Sujaya, Rupali, Abhigna, Aman, and all the team members, for this really decent initiative, for knowledge sharing to our young researchers, next generation of scientists coming up. My high appreciation. And I saw earlier speakers also, you have been trying your best to get, the people who can share their experiences and share the knowledge. Thank you very much for inviting me for this particular topic lectures, which is very favorite to me, chickpea, because, I spent my almost more than 30 years on this. So in this topic, I'll give an overview of the advances that have taken place in chickpea and the impacts of improved varieties in selected countries. So what I have done is that in this presentations, I have divided in different sections. First, introduce chickpea, because maybe there are many audience. I can see 30 countries and more than 4,000 participants there. So introduce chickpea. And then, because I will be presenting most of the work from ICRISAT, I will introduce ICRISAT there, and then give highlights of the advances in chickpea improvement, and then impacts of improved varieties on chickpea production, and then finally sum up. So to start from chickpea, this is a legume crop, which is the second largest grown pulse crop or food legume globally after common bean. It's a self-pollinated, diploid. Cool season food legume, and with relatively moderate size of genome, 730 megabase pair, and it's a cool season crop. It's in the semi-arid tropics. It's grown in the winter season, and in Mediterranean and temperate environments, it is a spring and summer crop. And because of its high water use efficiency, mostly this crop is grown as a rain-fed crop without supplementary irrigation. So large area is still under rain-fed situations. And being a legume, it has ability to fix nitrogen, biological nitrogen fixation, and it can meet up to 80% of its nitrogen requirement from BNF. And it's a nutritionally very rich crop. high in protein content, 20 to 22%, which is very high quality. Digestibility is very high, and rich in fiber and minerals and beta carotene. And we have two distinct types of types of chickpea, Desi and Kabuli type. Desi, you have almost 80% of area is under Desi, and about 20% is in Kabuli type. Desis are brown seeded or mostly, but they are green and black also. But Kabulis are white seeded, thin seed coat, and usually larger seed size, and always have white seed, white flowers. Kauli, but Desi can have white to pink, any. And chickpea has diverse uses. You can start from green leaves. You can eat as spinach. They are, nutritionally even better than spinach. You can use them like spinach. Green immature seeds can be used, dry seeds, and splits, flour, and then the remaining plant parts, fodder and feed, is very valuable. And if chickpea is grown in about 50 countries, and if you see that India has the largest share in chickpea production, about 69% of the production comes from India, and the remaining countries, none of the countries contribute more than 4% of the production. But in remaining countries, the major countries are Turkey, Myanmar, Russian Federation, Australia, Ethiopia, and then USA, Pakistan, Mexico, Canada, Iran, Argentina. So, and if you see that recent years trend in past 30 years, there has been a good progress in chickpea production. And almost in 30 years, the chickpea production has doubled globally because of 43% increase in area, and remaining because of the productivity, and because India has the largest share. The global trend is the mirror image of India. So in India, you can see in 30 years, 124% increase in production because of 52% increase in area, and remaining because of the productivity. In the rest of the world, the progress has not been that much, but still you can see 76% increase in production and 26% increase in area. Now, if you see the countries, and if you see the change in areas, of course, there is a big change in India. In last 30 years, there is a 52% increase in area in 30 years. And there are other countries, though they have less contribution, less than 4% in production, but you can see that there are many countries where the chickpea area has increased considerably in recent years. Russian Federation, Australia, USA, Myanmar, Canada, Ethiopia, Argentina, in all these countries, the area has gone up. But there are countries, traditionally, the area was high, like Turkey, Bangladesh, Malawi, Pakistan, where there is some reduction in chickpea area. Looking at the import and export, still, the Indian subcontinent, which includes India, Pakistan, Bangladesh, more than 50% import is by these countries. Though, India, we are contributing to 69% production, is still, we had import. Of course, in recent years, the import has gone drastically down, but, still there is import. And then other countries. And now in recent years, the trade has been very dynamic. Like you can see UAE importing 5%, but this is, from there, it is being exported to other countries, like 3% is exported from there. So there is a trade dynamic also. And in exporting countries, Australia has the largest share, 37%. And in recent years, this is the change in recent years. Russian Federation has emerged only in recent years as a major exporter of chickpea. India is doing very well and has started exporting chickpea now, among third the largest exporter of chickpea, India. Then traditionally Mexico has been exporting. Then we have USA, Argentina, Canada, all these countries. And developing countries like Myanmar, Tanzania, Ethiopia, Sudan, they have also started exporting countries. Now, point here I want to make is that as compared to earlier, now in recent years, we have more countries, for exporting chickpea than used to be 30 years back. And next section is change in scenario of chickpea cultivations. There has been a big change. As I said in the beginning, chickpea is a cool season crop, earlier confined to cooler environment, like in India. If you take its large area was in northern India and central India, but during the Green Revolution, chickpea was replaced by wheat and other high value crops, like later on mustard, rapeseed and mustard. So what has happened in India during the Green Revolution? We lost almost 3.6 million hectares chickpea area, 3.45 million hectares chickpea area from the north. But luckily, we had varieties which were short duration and suited for short season environment that helped in expansion of area to central and southern India. So in central and southern India, from 2.2 million hectare, the area increased to 7.1 million hectare. That was a big change in the chickpea cultivation in India. So chickpea moved to, from a longer and duration environment, cooler to warmer and short season environments. Similarly, in Myanmar, which has growing conditions similar to southern India, very short season environment, chickpea area has increased drastically, and this is also because of the availability of short duration varieties suited to this kind of growing conditions. Another big change we see is a wide range in sowing time. Earlier when we used to study, we used to study that, the best time for chickpea planting is, September to October in India. But nowadays, you see, the farmers are planting chickpea starting from September to December, and this is because the farmers, they want to increase cropping intensity. So they are doing their best to take, a crop in the middle after rainy season crops. And this is the picture I took in Andhra Pradesh, southern India. Very amazing. We were traveling and we saw this field and we stopped. We don't know, in fact, these were 3 farmers field or 1 farmer field, but very interesting. You have a crop here that is maturing. You have a crop here which is close to maturity, and there is another crop which is at seedling stage. So interesting, the kind of variation in sowing date. The another thing which is making this change is expanding area under rice fallows. The harvesting of rice is delayed, so chickpea is planted very late in November and December. So all these changes, the chickpea breeders have to keep in mind in developing varieties. Also, the climate change, climate variability. We see frequent changes in temperature and gradual increases in temperatures. So we have to keep in mind and develop the varieties which are more climate resilient. So in the breeding program, we have to keep in mind this changing scenario of chickpea cultivation, the area expansion in short season environment, and the variation in sowing date, and the climate variability. Now, coming to the next section, because as I said, I have worked more than 17 years at chickpea breeder at ICRISAT, and most of the work I will be presenting from there. I should introduce ICRISAT. People should know ICRISAT is one of the 15 centers of CGIAR, and it is among the first centers established by CGIAR in 1972. So these are the 15 CGIAR centers, and only ICRISAT is had headquarters in India. ICRISAT has headquarters in Hyderabad, in suburb Patancheru, and this is a beautiful campus, 1,400 hectares land, and very good landscape here. 2 types of soils, black soils and red soil here, and all the facilities you need for crop improvement research. And ICRISAT has almost 50% work in Africa and 50% in Asia. In Asia, only we have one, which is headquarters, 1 station, and of course licensing officer in New Delhi. But in Africa, there are 7 research stations, 2 regional hubs, and other research stations in Africa. ICRISAT has 6 mandate crops, global mandate for the improvement, 3 legumes, chickpea, pigeon pea, groundnut, and 3 dryland cereals, sorghum, pearl millet, and finger millet. And ICRISAT developed improved breeding materials and then supplied to the breeding programs globally, both private sector, public sectors, and these remains in public domain. So these are freely available to all. ICRISAT does not release any variety or hybrid, only supply the breeding materials, and the recipients, they evaluate them and then release further. So globally, 1180 varieties and hybrids have been released in 81 countries from the breeding material and germplasm supplied by ICRISAT. And this particular topic is for chickpea, where 181 varieties have been released in different countries from the material supplied by ICRISAT. Now, the ICRISAT, being a CGIAR institute, has a strength in the chickpea improvement program. It's the global program. And the biggest advantage ICRISAT has is the gene bank, where you have more than 20,000 accessions, the largest collection of chickpea germplasm available with ICRISAT, 20,000 from 60 countries. And then, all the facilities, Center of Excellence in Genomics. My colleague Rajiv Arsne, who spoke earlier, he is the, the director for Center of Excellence in Genomics established there. Excellent work, done in genomics and chickpea genomics. And then we have a platform for translational research on transgenic crops, a separate one there. And then all the facilities, for the phenotyping, like LeGI-Scan phenotyping platform, control environment facilities available, precision fields. And one, big advantage ICRISAT has is the global network of research collaborators. So now, coming to the breeding process itself, I must appreciate, my previous breeders there. They have been, very, I will say that, always considered, to improve the efficiency of the program. Highest emphasis on that. And, all the crop breeders, they know these breeders' equations that, the response to selection or genetic gain has these components, selection intensity, selection accuracy, and the genetic variability, and years per breeding cycle. So I see that from the program, ICRISAT's program, that there has been continuous efforts to enhance the efficiency of the breeding program. So the program has given emphasis on mechanization, in improving mechanization, automation, so that you can have precision and evaluate large populations. High throughput phenotyping, again, selection accuracy can be improved, precision of fields. And then in recent years, integrating other tools and techniques like marker-assisted selections and genomic selections in the program to improve efficiency. And very recently, highest emphasis on, digital data capture and data management, breeding management systems, so that you can have informed decisions in, selections and promoting your material. Enhancing genetic variability. I mean, the genetic gain depends largely on, what is the genetic base of your populations from where you are making selections. For that, the careful selection of parents, crossing designs to, combine favorable genes for the targeted traits, and suitable crossing methods for that. All these, and recent years, like using MAGIC multi-parent advanced generation intercross method to accumulate genes. So, over the years, ICRISAT program has, taken care of these and improved efficiency. And one area where I'll say that ICRISAT was pioneer was in rapid generation advancement and speed breeding. Because you can see the, the equations, the breeders' equations here, the T, is very important. If you can, have more generations per year or more years, more generations per year, you can enhance genetic gains, right? Substantially. So this is the one way of enhancing the, I will say, the most effective way of enhancing genetic gain. At ICRISAT, from the beginning, the breeders, they tried to take, more generations. When the program started in 74, normally there is one crop per year, but, you can take one crop in off season, where you have rain shadow area, you can take it in spring and rainy season. So they were taking 2 generations. But in as early as in 1981, ICRISAT, published this paper, field crop research, on rapid generation advancement, where they use the photoperiodic response and provided extended photoperiod to take up to 4 generations per year under controlled environment. So rapid generation advancement protocol was published for chickpea as early as in 1981. And that has been used very regularly by ICRISAT. The limitation is that, in greenhouse, you can use only a grow limited population. So it was restricted to selected crosses and developing mapping population like recombinant in red lines, but could not be used for, the remaining program because ICRISAT has a large program. Initially, they were developing like 1,000 crosses per year, later on 250, and now about 150 crosses per year are made. So it's not possible to take all the crosses. So later on, another, procedure was developed where you can take 3 generations in the field. So there is no restrictions on number of crosses that you can advance. So you can take 1 generation in a plant early, like September, and harvest it in January, February, and take another crop after February in the same, same location in Patancheru, and harvest it in March, April, May. And then your third crop you can take in the off-season nursery, which is, we found another location in Karnataka, Hiriyur, which is, better location than northern India, more predictable environment. So you take 3rd generation there in rain shadow area. So 3 generation in field without any, providing photo period or anything. You can do machine planting, and there is no restriction of area and land. So this we have been doing there, routinely taking 3 generations per year. And recently, we developed a protocol where, we combined 2 things. One thing is that, that was reported by Sethi et al. In 1981, extended photo period, and then combined it with, growing immature seeds. You combine these 2, and then you can further reduce the, generation cycle time. So you can take 7 generation for very early material and up to 6 generation for other, medium maturing and late maturing varieties. You can take 6 generations under controlled environment conditions. And this doesn't require embryo culture or anything. You can just plant immature seed on Petri plates, germinate it, and then transplant it. So this, again, we are using now routinely for for developing mapping population. And now, some crosses can be advanced using this. Now, coming to the next section of my presentation, which is on, the advances in highlights of advances in chickpea improvement. And the one area which has given, I will say the highest dividend is the development of early maturing varieties in chickpea. And ICRISAT has paid attention from very beginning, and it has location advantage also because it is in a very, short season environment there. So breeding for earliness is, very ideal environment for selecting for earliness there. So as I said earlier, that, chickpea is largely grown on residual moisture, and end of the season, there is a terminal drought. And also, as planting is delayed many times, so you get heat stress. And also in late sown conditions, like in rice fallows, you get heat stress there. And also we are seeing this climate variability. So crop is exposed to high temperature many times. So drought and heat have become very important. Terminal drought and, reproductive stage heat stress, they have become very important in chickpea. And for that, earliness is one way to escape these. And in chickpea, you have now, lines which are early 90 to 95 days, extra early 85 to 90 days, and even super early 75 to 80 days. Now, earlier, there was a debate that earliness will lead to, lower vigor and lower yield. But chickpea being an indeterminate crop, indeterminate growth habit, doesn't have that much penalty on yield as you have indeterminate crop. In breeding earliness, because the crop has plasticity. If the crop get favorable environment, like there is a rain or you provide irrigation, it can expand the duration because of indeterminate growth habit. So it doesn't mean that, earliness means the low yield. You can have, very high yield potential also in early background. So several high yielding early maturing varieties have been developed in chickpea, and this is the reason that in India why we were able to expand cultivation to central and southern India. And this is one variety, JG11, which is brought revolution actually in chickpea cultivation in southern India, particularly in Andhra Pradesh. And a lot of work done on understanding the genetics of earliness in chickpea. We have now better understanding of the genes involved, different genes, non-allelic genes. So you have the opportunity of selecting the genes based on their linkage relationship with other traits. These have been mapped also in different linkage groups. So you can select the gene depending on their relationship with other traits. For example, their linkage with resistance to ascochyta blight. There is a gene that is very closely located where you have there is a correlation with ascochyta blight. So you can have another gene which is located elsewhere for earliness. So you can have very precise breeding for earliness in chickpea. Coming to the next topic, this is a drought tolerance in chickpea, because the crop is still grown largely rain-fed without supplementary irrigation, and the terminal drought is the major constraint. Lot of work done on finding traits contributing to drought tolerance, but I must say that there is not much agreement on which traits breeders should use in selections. So breeders still continue to select for grain yield under master stress environment. A lot of work was done at ICRISAT in screening germplasms for drought tolerance, and there were some germplasms identified as having very high drought tolerance, like ICC 4958, and these were then again critically examined for the traits and found that the ICC 4958 drought tolerance is because of its vigorous root system. So it was studied extensively, and it was found that it's not only the shoot bigger, it is early root, not only the shoot bigger, but also a root bigger, but also shoot bigger. So that contribute to the drought tolerance in this, and this has been used in breeding program extensively. And selection was done again for grain yield, not for the root traits, because it is very difficult to select. And there are varieties released using this drought tolerant genotype. Later on, we decided to develop mapping population, for the root traits, because you can imagine how difficult it would be to select for root traits. So we took two contrasting parents, and in fact, developed two different mapping population. 4 parents we selected, no parent in common, and differing in their root system. And then analyze this. My colleague, working Rajiv Varsane with us, together, we were able to identify a genomic region, which Raju named as a QTL hotspot, which contributed to these traits, root-related traits, but also many other traits which were related. So these, interestingly, in different mapping populations, same genomic region we found, and later on, our physiologists, they worked further, and they found that even the other vigor traits, they were mapped in the same genomic region. So this genomic region is for the vigor traits, which contribute to root vigor, shoot vigors. And then later on, we introduce and progress this genomic regions to many varieties, and at least one introgression line of JG11 has been released in Ethiopia recently as Geletu. There, this drought tolerant variety, you can see these introgression lines, the line and the variety and introgression line, the variety JG11 and its introgression line here. So this has been released, and also ICRISAT worked in close collaborations with Indian Agriculture Research Institute, IARI New Delhi, where Dr. Bharadwaj, he introduced this genomic region in a popular variety Pusa 372, and then a variety has been released recently in India, the introgression line of Pusa 372. So there are at least 2 varieties released by introducing this genomic region in chickpea, and many are in pipeline. Heat tolerance is a very interesting topic. Earlier, we thought it is a cool season crop, may not have that much variation. And when we started taking 2 generations at Patancheru, and the second generation was always subjected to, a heat stress, at reproductive stage, the temperature was like more than 35 degrees Celsius. So we saw big variations there for heat tolerance. And normally optimal temperature for chickpea is 10 degree to 30 degrees Celsius. And if the temperature increases beyond 32 degrees Celsius, you see a drastic reduction in, pod set and yield in chickpea. So what we did there, when we saw that there is a large variations, we, screened the reference collections, and then we found that, there are big differences in chickpea for heat tolerance. And we developed a very simple screening method. Just, the delayed planting, you find the time that, your reproductive stage coincide with high temperature like 35 degrees Celsius. So luckily at Patancheru, you get very predictable environment. If you plant in February, the temperatures will be, 35 to 40, 42 degrees Celsius during reproductive phase. So you can screen. And we found that you can see here, the heat sensitive line will grow normally if you irrigate it, and it will produce flowers, but not produce any pod, no pod set. If you get some cooler time, you can get some pod set. And there is a big variation. You can see the parallelly growing, drought tolerant, heat tolerant line and heat sensitive line. Heat sensitive line, no flower, no pod. Flowers are there, but you can see the normal podding in heat tolerant line. And so you can use simply this, the filled pod number, filled pods per plant as criteria for selection for heat tolerance. Even you can do visual selections. You don't need any sophisticated method for heat screening. So, this really helped this large variations in, progressing for heat breeding for heat tolerance in chickpea. Now we have, many varieties, many breeding lines for heat tolerance in chickpea. Particularly, I would like to mention one line, ICCV 92944, developed by ICRISAT. It has been released in 4 countries. In India, released as JG14. In Myanmar as Yejin 6, and also released in Bangladesh and Kenya. So it shows its, climate resilience and adaptability, wide adaptability. And we have conducted large number of experiments in India in late planting condition, and we found that this variety gave 10 to 25% higher yield than the best varieties available in these regions in late sown conditions. Now, heat tolerance breeding is under mainstream of ICRISAT chickpea breeding program. Means all the breeding lines which are promoted, they should perform well in both the normal condition and heat stress condition, then only the lines are promoted. So this is now in the mainstream of the breeding program. Also, we developed a mapping population. One of my PhD student, he worked on this mapping for mapping QTLs for heat tolerance. He found 2 major genomic regions for heat tolerance, which together accounted for more than 50% phenotypic variations. We had to work further, to validate these QTLs and markers to use routine use in breeding program. But there is a good progress in this direction also. And all efforts have been made from beginning to have multiple crosses and accumulate favorable genes. And recent years, this development of multi-parents advanced generation intercross populations. So we selected 8 varieties and elite breeding lines, the best cultivars, in fact, and made all possible crosses, excluding reciprocals. So 28 two-way crosses, then 2-way crosses, 4-way crosses, then 8-way crosses, and developed these magic populations. So it had 2 purposes: discovery. For that, we developed magic lines using single seed descent. 1130 lines were developed. They were sequenced and for gene discovery and mapping. And then we had parallelly for breeding this bulk seed, which we harvested from 8-way crosses. We bulked them, and then this bulk we shared with many partners, many breeding programs, and they selected further. We also selected at ICRISAT, and now there are many promising lines under testing and will lead to many varieties from this program. So we found that this multi-parent advanced generation intercross method is very useful for accumulating favorable genes for traits which are multigenic. Coming to the progress in breeding for biotic stress tolerance in diseases, Fusarium wilt is the #1 disease in dry areas, in warm and dry areas. Fortunately, here our pathologists that did a great job in developing screening methods. Very reliable screening methods have been developed, both field screening method and pot screening methods. And the sources of resistance. Several sources of resistance have been identified, and they are very stable sources of resistance. For example, in this picture, you have this is used normally as a resistant check. WR315 is was identified maybe 40-50 years back. Still it is stably resistant there. And then you have susceptible checks like JG60 too early wilter and 850 late wilter, and very good screening, perfect screening. You can just see in the field and just harvest the resistant plant. Sorry. And initially, we had, very few sources of resistance in Kabuli types. So ICRISAT used, Desi and Kabuli crosses to have resistance in Kabuli backgrounds. So drawback was that, because of Desi Kabuli crosses, the seed coat color in Kabuli had the, slightly brownish tinge, which reduces the market value of Kabuli type. So we thought, we should have good resistance sources in Kabuli types so that, we don't compromise on seed quality. So we screened a lot of material later on. And in large seeded Kabuli, we found sources of resistance like this one, particularly very highly resistant. And later on, we used only Kabuli sources for resistance, for wilt resistance in Kabuli, so that you can get very good seed quality. So now you have, this disease, you have perfect control. And this is a disease must to have trait in the release of varieties, means you can't release a variety unless it is resistant to Fusarium wilt in India. Also, a lot of work globally done on mapping genes for Fusarium wilt resistance, different races of Fusarium wilt, and some efforts on integration of genes using markers. At least there is one example where ICRISAT worked closely with in Indian University in Raichur, University of Agriculture Sciences, US Raichur there, for introducing Fusarium wilt resistance through marker-assisted back crossing in a very old popular variety, Annagiri 1, which was one time a dominant variety in southern India. Almost 90% area was under this variety, which later on was replaced by JG11 largely. So they introduced this Fusarium wilt resistance gene in Annagiri 1 and released recently a variety, Super Annagiri 1. So this is one marker-assisted variety for Fusarium wilt resistance released in India. Of course, you can select simply by in the field screening, but when you are using this trait along with many other traits, marker will be useful. But only if you are doing for, Fusarium wilt resistance, I don't think there is a need to go for markers. You can simply screen in the field, but it will be very useful when you are handling several traits together. Ascochyta blight is, a foliar disease, which is a disease of cooler temperatures, and usually comes when there is a cloudy and wet conditions, growing conditions. This disease in India does not come in central and southern India. It's only in some pockets in northern India, but globally this is the most important disease. In Australia, Canada, USA, European countries, most devastating disease. And this is, pathogen is highly variable, evolving pathogen, and resistance is quantitative. So it's not as easy as breeding for Fusarium resistance, and you don't get that stable resistance also in case of Ascochyta blight. So, and breeding for like institute like ICRISAT is challenging because you don't get disease in the field in southern India. So you have to have a control environment facility for screening. So pathologists, they did a great job in developing these facilities. This is a chamber where you have a perfect control on humidity and temperature, and you can inoculate and give the required temperature and humidity for Ascochyta blight and screen the material, large material at seedling stage. If you want to screen at adult plant stage, you can bring the branches from the field and then you can screen for that also. Then what we used to do is you used to screen bulk F4, F5 at seedling stage, transplant the resistant plant to greenhouse and finally to field. And then the progenies is screened in the hotspot locations in northern India. Punjab Agricultural University, Ludhiana, very good, location for screening for Ascochyta blight resistance. So this is the field from there. So you screen there finally, and then you can promote the material. And so there has been globally extensive efforts on breeding for Ascochyta blight resistance. And we have, good level of resistance, not as high as fusarium wilt resistance. You don't have immune varieties, but you can have, if you take 1 to 9 scale, up to score 3 resistance is now available in varieties. And this has been possible by, intercrossing resistant varieties, accumulating favorable genes. And this is one example of a variety developed, the line developed by ICRISAT, Desi Chickpea line, and released in Ethiopia. You can see the level of resistance for Ascochyta blight there. These are susceptible, and this is the line. So, and also efforts on, lot of efforts on mapping QTLs and some efforts on, testing for whether the markers are working efficiently or not, have been done. We were not very happy with the efficiency we got, so still there's work in progress for, fine tuning that and finding, better markers for Ascochyta blight resistance. Another disease is dry root rot, which is a disease of, dry environment. Very warm conditions and high temperature. So this is now the because fusarium wilt is under control, so this disease became a devastating disease in dry areas like central and southern India, Myanmar, and even African countries. Unfortunately, we don't have very high level of resistance available for this disease. So the best possible at present is maybe the score 5 in 1 to 9 scale. The varieties. So we have to, settle on or on moderate level of resistance available, and then, use, management to handle this disease. So there is, good screening methods available, both, control environment and field screening, and but, we can have varieties only up to the score of 4 at present. Pod borer is the most devastating, the pest of chickpea. And again, for pod borer, we don't have very high level of resistance available in cultivated germplasm. You do have some variations when you grow together, preferential difference you can see, but when you grow a sole crop, Helicorpa will eat in any way, everything. So the only hope we have is, the transgenics and some hope from wild species, and the works is in progress for this. And in near future, we may have, tolerant varieties coming from transgenics or maybe from wild species in the future. And it was mentioned in introductory remarks that, in developing countries, the mechanical harvesting is needed by the farmers now. Chickpea in all developed countries is harvested by machine only, like Canada, Australia, US, or European countries. There, they select for the plant type which are amenable to machine harvesting. But in developing countries, the plant breeders, chickpea breeders did not pay attention because it was not a priority trade that time, plenty of labor available. So, unintentionally, the selection was, for the bushy type plant type. So most of the varieties, released are like this type, bushy type in India and other developing countries, which are not amenable to machine harvesting. So you need a plant type where, you have a semi-erect growth habit, and the first parts is like 20 to 25 centimeter from the ground. It's not a difficult trait to breed. Lot of variability in germplasm. You have to just select for it, and you have several advantages, in breeding for machine harvestable varieties. And in fact, this is the need of the hour. The future is mechanization. In the future, chickpea will be harvested only by machine like developed countries, in developing countries also. So there is a lot of progress in recent years. We had a laws network program in India. Many universities, ICR institutes were involved in that, and very good progress made in few years. 4 varieties have been recently released, machine harvestable, and many in pipeline. And I'm very confident, in chickpea in India and in other developing countries in coming years, chickpea will be largely harvested by machine only. Another trait which is important, for labor saving is, herbicide tolerance. I know I'm coming from Madhya Pradesh, soybean crop, how tedious it was earlier when, it used to be, the weeding was the biggest problem, and manual weeding used to be there. And for farmers, most expensive operation, weeding. And now soybean, weed management is only through herbicide. Harvesting also mechanized only, So same thing in chickpea in the future, it will be, the weed control will be through only the herbicides and machine harvesting. So lot of work in recent years is developing herbicide tolerance. We screen large material, find good variations. Like you can see this variation for M.M this is resistant variety, susceptible. Still, we have to do some work to have varieties, because when we spray herbicide, we are seeing that, in this particular case, the tips are damaged and maturity is delayed. So at lower dose, there is no penalty on yield, but in short season environment, delayed maturity is not desired. So there is still, we need to do some work, but I'm very confident the way we are moving forward in future, we will have, very good herbicide tolerant varieties in chickpea in these countries. And also, work efforts on mapping for herbicide tolerance. One of my PhD students worked on it and identified, some QTLs for herbicide tolerance, which need to be further validated for use in breeding program. Also, if you see, the breeding programs have taken good care of the market preferred grain trait. I have seen the program evolving. These are the kind of varieties we had in the beginning for in Desi type, and now you can see that most of the varieties are this type, lighter brown color and more plumpy than, very wrinkled type of seeds. Similarly, in Kabuli type, earlier varieties were small seeded Kabulis, 25 grams to, then we have, large seeded about 35, 34 gram to, varieties, and extra large Kabulis in recent years, above 45 gram, 50 gram, 55 gram per 100 seed weight varieties. Now plenty of, and this is how, like India has progressed now. And in India now, India used to import, these extra large Kabuli earlier. Now India is exporting, to other countries. In fact, the Indian export of chickpeas, mainly the Kabuli types, India is exporting. So excellent progress in this area has happened in recent years. And another area which has tremendous potential, where the work has not been that much in chickpea as compared to many other crops. And this is the one area where a lot of progress will take place in coming years. Unfortunately, in Harvest Plus, when CGIR started, somehow chickpea was missed from the crops included in Harvest Plus. Lentil was there and many other crops were there. And good progress made in all those crops which were included in the Harvest Plus program. Pearl millet, for example, is a crop, a model crop where, so much work done on improving, iron and zinc, that India now has minimum standard for iron and zinc for release of varieties, and, I had a dream that in chickpea, we should reach to that level, So there are opportunities, there are good variations for protein content in chickpea, 16 to 28%. And I believe that, we can at least, 25 to 23%, we can increase protein content in chickpea. And iron and zinc, we can increase up to 50 to 100%. So these are the areas where, work has started, many labs are working, and in the future, I think we will have varieties which have, higher content of protein and, iron and zinc there. And, why not? We should mainstream these, if the consumers can, from the same quantity of grain, if they can get, like 25% higher protein and maybe 50% or 100% more iron and zinc, that will be, beneficial to their diets. So, these traits should be given, highest emphasis in the breeding program. Now, coming to the last section of my presentation, the impact of improved varieties. I have taken some example. Globally, I have already shown you that, there is almost the production has doubled, and 50% came from area expansion, and almost 50% from the, productivity enhancement. So the varieties have played a very key role, and particularly in expanding areas in, new niches like short season environments in India, like southern India, Myanmar, expanding area to these, short season environment. And this has been possible because of the development of short duration varieties. Now, take India. In last 3 decades, chickpea production in central and southern India increased 3.7-fold because of 2.2-fold increase in area and 1.7-fold increase in yield. And this has been possible because of only the suitable varieties, which were available for this thing. You can see the graph here. And ICRISAT with, partnership with Indian program has played very important role, because in India, the varieties released from, ICRISAT supplied breeding materials in partnership with, ICRISAT program, they contribute in past 10 years to 48% of the total indent of chickpea breeder seed in India. And if we take central and southern India, 64% of the indent of breeder seed in central and southern India is ICRISAT related varieties. So I must say that, there has been a significant contribution of the partnership of, Indian National Program and ICRISAT in chickpea improvement. And if you compare central and southern India with rest of the world, these are the graph at the same scale. The kind of progress made in central and southern India in past, 3 decades is, greater than the progress made by rest of the world, excluding India. You can see that, in central and southern India, the production increased from 1.9 million tons to 7 million tons in, past 3 decades, whereas the rest of the world, 2.6 to 4.3. Area, you can see 3.1 to 6.7 as compared to the rest of the world. And productivity also, increased very much, 610 to 140, 1040, more than 1 ton per hectare. So you can see that central and southern India is remarkable progress, and it's mainly because of these short duration varieties. And now, if you take 2019, chickpea production by central and southern India is 62% higher than the rest of the world, excluding India. So chickpeas, that much chickpea is being produced in central and southern India. So that's the big impact of improved varieties. Of course, the production technologies goes along with that. Myanmar is another, example where, a revolution has taken place in chickpea. And, Myanmar is a country where the agriculture department is maintaining variety-wise statistics. So I used to get regularly their variety-wise area. And you can see this is the area, during 2019 to 20. These varieties, 6 varieties, they account for 90% of the chickpea area in Myanmar, all released from ICRISAT bred materials. There in Myanmar. So 90% area is under these short duration varieties. And you can see in this, 20 years, past 20 years, the production increased 325%, 325%, increase in production. Area 131%, and, yield, you can see 715 to 1.3 ton per hectare. So there is a revolution in Myanmar, and Myanmar has started exporting chickpea because of this. Another example I have taken is Ethiopia. In Africa, this is the one country where the production is the highest, is Ethiopia. And where, you can see the production has increased drastically, and their production is not because of the area. Area didn't increase much, only 13% increase in area. But the yield, you can see, from 891 kg to more than 2 ton per hectare increase in productivity. In fact, this is the one country where you have the productivity very high, 2 ton per hectare. And we were, ICRISAT has been associated with Ethiopia because our presence in Ethiopia. In fact, more than 80% of the area is under improved varieties, and these varieties, ICRISAT related varieties, if you take, there are total 31 varieties released in Ethiopia, 21 are from ICRISAT bred materials. So ICRISAT has a big contribution in chickpea revolution in Ethiopia. These are some examples, where the varieties and production technologies had made great impact. Of course, there are many others, like Australia, where there is a revolution because of the development of ascochyta blight resistance varieties and production technologies. And there are many other cases, but these are three big examples. Coming to my last slide, summing up, I think you will agree with me that development of high yielding short duration varieties with high resistance to fusarium wilt and improved tolerance to terminal drought and reproductive stage heat tolerance helped in expansion of chickpea area in new niches, as you can see in India and Myanmar. And adoption of improved varieties along with improved production practices helped in increasing yield levels of chickpea, as you can see many examples. And opportunities exist for further enhancing growth of chickpea production by improving both productivity and reducing yield gaps and expansion of area in new niches. Still, we see large rice fallow areas where there are opportunities of expanding chickpea cultivations. And yield gap, means promoting the already available technologies that will lead to enhancement of productivity. Excellent progress made in improving heat tolerance or suitability to machine harvesting in recent years. And I believe that these traits can now be in mainstream of the breeding program, means all the materials which are being developed should have suitability to machine harvesting and heat tolerance in there. And there are areas where we need to do little more work. Efforts should be enhanced in increasing protein content and micronutrient content, and also we still need to do more work in herbicide tolerance. And we are now in a better position as compared to earlier. We have access to novel tools and techniques like genomic resources, precision phenotyping, rapid generation advancement protocols are available, which should help our breeding program if we integrate these in having greater efficiency in our breeding program. So this is what I had, for this presentation. And I think, yeah, thank you very much. Yes, over to you.
Hello?
Yeah.
Yeah, please. Maybe I can stop sharing now screen. Yeah.
Hello?
Yeah, please.
So thank you, Professor Gaur, for an enlightening talk on chickpea improvement and production. You have been a pioneer behind chickpea research and production, which was an orphan crop legume 2 to 3 decades ago. We are grateful to you for bringing in such an important nutrient-dense legume to our table. Your talk shed light on significant aspects to consider in chickpea breeding. This is evident from great responses received on our YouTube channel. We have many interesting questions, and are you happy to take a few of them?
Sure,
from our
sure.
attendees?
Sure.
Thank you for that. I have picked a few questions. Due to time constraint, we are unable to answer all of them. So please accept our apologies for that. So the first question goes, do we have any gene editing success stories in recent times in chickpea?
I'll say that, the work is in progress. Still, ICRISAT has started, and many other institutes have started work on gene editing. I think we'll have to wait some time for, having results out of these, but this is one area which has high potential. And I'm very confident, that, this is another tool in our hands, which will, expand our ability in chickpea improvement. So we had to wait some time for, having a good successes. Yeah, but it will be.
Yeah. Thank you. Thank you for your response. You mentioned that you are targeting herbicide tolerant cultivars. So what group herbicides are you focusing on in chickpea? So what are the important herbicides?
Yeah, so we, worked on, 2-3. Imidazole is one, metribuzin is another. There are other groups working, like Canadian groups are working, and others, Australians are also working, for different groups. This is the area where we started only recently. This is one area where we have to do more. So we focused metribuzin. We didn't get much, the variation, that level of tolerance as we got for imidazole group. So we focused mainly on that. And I see that there are opportunities in that area, but we must not forget, we should work on others also, where we get, less toxicity on the crop, and you get, better control of herbicides. I think, this is another area which I will say that in few years you will get more results coming up, and we will progress like any other crop.
Yeah, certainly we always aim for less chemical residues in crop. And I do understand, because I had been in the same field. Okay. The third question goes like, what is the current scenario of MAGIC program in chickpea, and how many crosses are made, and up to what generation it's been progressed, and when the selections are made?
Yeah, so
this particular thing.
So I mean, there are now many other institutes are working on MAGIC. We started long back on. We started with one set in Desi type. That was the beginning, which I presented. It was published also recently, and then we started on a Cobbly MAGIC lines that was also developed there, and now there are others in pipeline. For MAGIC. So I will say, to all the breeders, young breeders who are, intend to start, this is one potential area. It requires actually extensive work on crossing. So it's tedious in the beginning, so you have to have very skilled manpower for making crosses, So you can take 6 parents or 8 parents, depending on your need. And then you can, if you want to have for discovery, then of course you have to go to the reed single seed descent, after once you have 8-way crosses, and you have, you can go single seed descent, develop lines, and then they are just like, your rills, you can evaluate them and for the mapping. But for the breeding program, you don't have to worry. You can bulk at that stage and take the bulk from, your F2 to maybe F4, F5, that bulk, without even any selection. Even if you are doing selection, select for simple traits. So take that up to F4, F5, and then that bulk is, for you a gold mine. You can then, evaluate for different traits. So what we were doing is that, we developed bulk quantity of F4, F5, like a 70-80 kg, and we distributed that bulk to many programs, and then they, screen that. So you can do that. So 2 ways of doing it. You can have, early selection for simple traits if you want, or you can just progress up to F4, F5, and then you can make selections in that.
It's all about chances, you say. Coming to fourth question. When we speak about high throughput phenotyping, what are the automations included for a large broad acre field?
Yeah. So here, high throughput phenotyping, in the sense we have taken is, both where you have even the field one, where you have actually the large population precisely you can screen, So those are also there. And even I will say fusarium wilt resistance is, you can have hectares and you can have very precise. Then coming to lab-based, like now for iron, zinc, protein, it's all standardized, you can do that, NIRS-based and XRF-based, you can do that. But still, and some in physiology, but it will take time. little time.
Yeah, like phenotyping, I mean to say, like there are automations like NDVI sensor,
Yeah.
and what other technologies are used in recent times?
So for us, we have, only what we have presented is still in physiological traits. We haven't done much, in that, because as I said, there is still, if you see literature, there is a very diversity in opinion that this trait works in certain conditions, like even for root traits, you will find papers that say, root trait doesn't work everywhere, in certain environment only, like southern India, where you have a very short season environment, terminal drought. It works. It doesn't work in northern India, actually. So, so for that, we haven't used much, but for other traits, we have been. And now for,
it's kind of,
quality,
it's kind of subjective. I get your point.
Yeah.
So this is our final question. What is the status of chickpea in terms of grain quality, such as beneficial carbohydrates? How far chickpea has progressed as a functional food?
Yeah, this is, of course, a very potential area, and some labs did a lot of work, like in Saskatchewan. Ravi Chibbur's lab were working, on chickpea nutritional quality. In Australia also, some of the work there. So there is, work on carbohydrate also, amino acids also. There is some work, and now, also efforts on, in fact, the protein as such, extraction of protein in that area also there. Work on fluorescence also, RFOs, raffinose family of oligosaccharides, in that area also. There is a variation, there is work, but it's not that problematic situation in this legume as in others, so and there is diverse opinion also there, whether they are useful. In some cases people say they are beneficial. So there is, but that's all potential areas there, industries areas. I will say in chickpea, as compared to many other crops, there has been less work. This is the area, nutritional quality and functional food area is just the emerging area for chickpea.
Okay.
Where you will see the progress.
Yeah. Okay. This was the final question. Thank you, Professor Gaur, for your responses and valuable insights. This is the end of a questionnaire session, and I now request Abhigna to take over the session. Thank you.
Yes, ma'am. Well, that was extensively informative. Thank you very much, Professor Pooran Gaur, for such a flawless progression of event and for illuminating the subject to this extent. Thank you very much. For the next event, I would like to call upon Dr. Sujeshri, Chief Operating Officer of Plantgenomia from Indian Agricultural Research Institute, New Delhi, for carrying forth the interview round. Over to Sujeshri, ma'am.
Thank you, Abhigna. Thank you, sir. Thank you for your really fascinating and informative presentation. And your articulation of your all research results were definitely a valuable asset to the scientific community. So, sir, I have a few questions to you that audience are really curious to hear from you. Sir, initially, my first question is, sir, we are very excited to know that what does it take you to become an accomplished scientist? The journey to the success. I mean, you can share your earlier career goals, the hardships, and the memorable moments and the turning points in the journey. So please share with us your winning formula.
Thanks. I mean, this is something, I have shared with other platforms with the students, interacting with them, because I had a long journey, and, maybe some students will get encouragement from that, because I was born in a village in Madhya Pradesh, a small village, and where the school was only up to, primary, 5th standard, and, that time, it's government school only, of course, and then you have one school, one room, one teacher up to, 1st to 5th class. And then, you can imagine how though the teacher was, very good, but how much he can pay attention to the students, So the journey started from there. And then, after 5th class, you have to go to another village, walk 3 kilometers, to go to another school, middle school. And just imagine, no road in rainy season, you can't even have your shoes. You have to put in your hand, shoes, and on the shoulder your bag, and you have to walk 3 kilometers in mud and go to the another school. So what happens, during that process, many of your colleagues, they remain there, they will not go there. So only some will go to that school, and unfortunately, no girl will go to that school. They will drop out at 5th level that time. So then you go to another school up to middle school. And fortunately, though that middle school was in a school, but had very high standard, in a village, government school. And that was, the beginning of my interest in education. The headmaster was so good, very dedicated teacher, and himself was teaching important courses. So there I developed a dream of becoming a teacher. So that was my, initial dream, that I should become a teacher. After 8th, then you have no other choice than go to town. And, in that age, you have to go to a town, and that time, no hostel, you rent a house, rent a room, and do everything yourself, cooking, grocery, everything, washing clothes, everything you do in a school, higher secondary school. And that time when we studied, it was higher secondary school, not 10+2. You had to, another thing I missed is that you had to select your subject in 9th class. So, you take the stream, whether you want to go to biology, mathematics, agriculture, or commerce or arts, you have to choose in 9th class. And once you choose, there is no going back. If you take, mathematics, you can go to engineering. If you take agriculture, you can't go to biology or, or engineering. You can't go to medicine. So, and in that time, selecting subject for a boy in 9th is a very difficult task, So you do whatever others are doing. So I took agriculture because, I came from a village. All others were taking agriculture. I also took agriculture. I didn't know anything actually, But, your luck, the school again, you get some good teachers, and there was an agriculture college in that town. And he used to take us for practical to agriculture college. And then I got impressed with the college. It was a very good college, nice building, very good landscape, and the professor's quarters fascinated me. And then I developed another dream, not teacher, I should become professor, in the university. So that I went to then agriculture college there in the same town, a small town, Hindi medium, even BSc in Hindi medium. And at that time, I never traveled to other places, So I was just studying. I saw only Sior and Bhopal, no other town that time, And fortunately, I must say that we have a very good education system in agriculture. When these universities were developed, they were developed in collaboration with US universities, like University of Illinois helped in developing JNKVB and Pantnagar universities. So we had the same, system, curriculum, and same system, very good education system, agriculture system in India. And they had this educational tour. So that was, breaking point, they took us to an educational tour. First time I traveled in train, went to Southern India tour, saw all these cities, and, also saw the headquarters of my university, and then got interest that I must go out of this town, go to other place. Then I went for master's in the headquarters of the university, which is Jabalpur, did master's there. And everywhere I will say that, faculty was good, they were all with good mentors, I was lucky. My professor, my supervisor was Dr. Swaminathan's student, Professor C.B. Singh. He returned from Australia, from postdoc, very energetic, young, energetic scientist, So he was my supervisor and a mentor. So, we studied there. And then after my master's, I got CSIR fellowship, started PhD there itself. Coursework completed, was my 3rd semester, almost close to examinations. And then, there were vacancies in the university assistant professor. And the qualification was master's degree. I applied for that and I got the job. So I was in a dilemma because my dream was only to become university professor. That's it. I had no other dream. So I thought, what to do now? Should I continue my PhD or should I join? So I made a decision to join. So I left my PhD that time, and then joined as assistant professor. And my professor, he knew my interest in teaching, so he gave me class from the next day. So I started teaching there, from the next day. So this is how I started. Then, I got stuck in the rule regulations. You can't do PhD in coursework unless you complete 5 years. Don't get a study leave. So I regretted. I should have completed PhD and then join. That would have been easy. Then I registered in a traditional university for PhD. Again, completed my research work. Then during that time, I realized I must go to other country for PhD. My supervisor also, he insisted me that you must go out. And then I started trying for a scholarship because I can't afford to, this. I need to have a scholarship. So I tried, 2 years, luckily got Commonwealth scholarship and ended in Canada there. And that time, I was really reading a lot that time. That was the era of protoplast fusion, the biotechnology. And I developed a proposal on transferring NIF gene from legumes to cereal. That was my PhD proposal for Commonwealth scholarship. So I went there, and then, in Commonwealth scholarship, they themselves apply for admissions, and you don't have a choice for supervisor, they decide, So I got a supervisor, and again, my luck, Professor Als Linkard, who is actually father of pulse industry in Canada. So he was my supervisor. So when I met him, he said, Pooran, you want to work on this proposal? This is not for PhD student, this is a, chances of success are very limited. These are for researchers, not PhD students. So I will suggest you to rethink, but if you insist, then I will find another supervisor for you because I am a plant breeder. I don't have expertise in this area. But when I came to know from others that you are very lucky you got this supervisor, then my aim was that I have to work with him only. So he gave me 15 days time to think of another proposal, So I again studied, and that time it was beginning of markers work, isozyme work. And I found that there is a lot of work in pea and other crops, but not in chickpea, no paper in chickpea. So I went to him that I want to work on isozyme gene mapping in chickpea. He said, wow, that's very good, So this is how I started working with him. That time, he was just experimenting on chickpea. This was not a commercial crop in Canada. And I must say that he was a very visionary scientist. He was hired as a pea breeder, but he's the only scientist working that time on pulses. He introduced lentil in Canada. He introduced chickpea in lentil in Canada and made that country pulse country. So he's known as father of pulse industry. So he had a vision. So at that time, he was just experimenting on chickpea. It was not a commercial crop. So, my another colleague was working on this interspecific hybridization. I worked on gene mapping, developed, the first gene map there. And because I went on a study leave, I came back to my university and then, requested my professor there that I want to continue on chickpea. And he was another kind person, who allowed me to work. He said, because we don't have a chickpea program in Jabalpur, a chickpea program is at research station, and we want you here at the headquarters. So if you want to work on chickpea, you have to bring your own projects and resources. So I started journey there. Then, that was again developed as a center there. I initially sub-center, main center for chickpea there. And then one fine morning, I get a call from ICRISAT that, can you come to ICRISAT for 1 year? Our chickpea breeder is going on sabbatical to Canada. Can you see the breeding program here? So I came to ICRISAT for 1 year as a visiting scientist and then ended up there, then continued there for almost 19 years there as a chickpea breeder. And finally, later on, I was never interested in management position, but you have to accept sometimes. Then, in last 2 years, I took management position there as a director of Asia program.
Yeah.
And this is how the
Yes, sir. It's very interesting and motivating. Yes, sir. I have another question. That is, as you told, sir, you are the director of Asia Research Program at ICRISAT. So we would like to hear from you the importance of leadership skills in scientific workforce.
Yeah, I think, this is very important. Once you grow, sometimes whether you like or not, you have to take a leadership role. Even you are a small team, even if you are running a breeding program, you are a leader of your program. And this is very important. And the key is that, having a respect for each team member, they're identifying their potential and, making that environment where everyone grows, everyone feels attached to that program. I think that's the key, in leadership role. You have to encourage people. For everyone has something unique. You have to identify that and then encourage them to use their full potential. And then, you will be, one day a very successful leader. Yeah.
Yes, sir. It's great. Great words and very insightful thoughts, sir. So my next question is, sir, it is well known that your project, that is ELSK, Extra Large Seed Kabuli project, it helped the country to shore up in chickpea production and also to go beyond the self-sufficiency of our nation. So, sir, what are your suggestions for having a scientific research program with greater impact, or which is having a greater impact in farmers' field?
Yeah. So, for specifically for plant breeders, you have to have a longer vision. One thing I learned from my supervisor, which I always remembered, my Al Slinkard, when I met him, that, Pooran, for becoming a successful breeder, you have to think 10, 15 years ahead. Because your varieties, you develop varieties, they will be tested, they reach to the farmers. It takes 10, 15 years, sometimes even more than that. So you have to think what would be the requirement, 10, 15 years ahead. That's the vision of the program you should have. And, it's all about, your creative thinking, and sometimes there will be hurdle. I can give you many examples. You will have hurdles. When I started in my university breeding program in Jabalpur, that time, there was no Kabuli chickpea area. So I thought that time, that there will be, if we give one thing, another thing for breeders is that your job is to provide choices to farmers, not to dictate farmers. They will decide. Your job is to provide choices. So I was thinking that time that, if the farmers, they get Kabuli varieties, which are as good as Desi varieties they are growing, why not they would grow? So I started a Kabuli breeding program in Jabalpur, and then we had a meeting. We used to have annual meeting, our review meeting, and my director research, when I presented, he scolded me, who asked you to start this program? What is the Kabuli chickpea area in Madhya Pradesh? I said, no. Then why you are wasting resources, when there is no area? So I thought, well, it's not his mistake, he's not that crop, expert, So I thought, okay, I will still continue, but, so I still continued that work. And because I knew that one day, and that you can see now, Madhya Pradesh is the largest, Kabuli producing state now. But if you stop that way, then, sometimes you have to realize that others may not have that level of thinking of your area. They may be expert in their area. We are not denying that, they are not expert. He was great in his area, but maybe not in this area. So sometimes you have to have that vision, longer vision of future. And always remember that, you are providing choices only. So you will have the hurdle. When we started extra large Kabuli chickpea program, it came from farmers only. We were traveling in southern India, and then we found some farmers growing extra large Kabuli varieties. They bought from market. India was importing extra large Kabuli. There was no variety released in India. So he was a progressive farmers going to supermarket. So he bought the chickpea. Why not to grow? So they were growing very poor yield, very long duration Kabuli, but good seed. So I saw that, I said, why not we develop varieties which give them, which are locally adapted and have the same grain quality. When we submitted that proposal, one big authority challenged it. He said, there is a negative correlation in seed size and yield. If you breed extra large cowbelly, then productivity will go down. We want to enhance productivity of the crop. You will reduce the productivity. And then he asked us, what is the advantage, nutritional advantage of extra large cowbelly? We have no answer. There is no advantage. Then he said, why you want then extra large cowbelly? Then we said, if you don't provide options to the farmers, they will be growing the varieties which are coming from import, poorly, adapted varieties. So our job is to provide them options. If they found it less profitable, they will not grow it. So he was convinced. And then we got that projects and, so sometimes, you have to use your logic. You have to, think that way, beyond, out of the box.
Thank you, sir. It is a very helpful comments from your side.
Yeah.
Sir, my next question is, we have research scholars and scientists as our audience. So they are eager to know, like, sir, when you choose some research project, while choosing a research project, what are the strategic approaches or the technical competencies you are looking into that?
Yeah. I think, this is a combination of, approaches. There is no simple answer to it. First thing I will put is the own interest. You have to see your own interest, what you like to do. Sometimes, that is very critical. Then, what is going on in the world, because there you will excel where you have interest. So first thing, you see your own interest, where you want to go. And then, of course, you have to see that which has the potential, which is the growing area, future potentials also. And nowadays, we are lucky, we are in the age of, internet, the informations, it was not available that time, in our time, we had the difficulty, I mean, see, poor data system for us to make a decision, go to the library, whatever. You get old journals, you don't get all journals, and you read them, and then you think, what you can do. Nowadays, you can have all informations available with us. So see your interest first, and then you see the areas which have, the potentials to grow, and then you can select. Also, the institutes where you want to go is very important. You have to do that, a lot of exercise on that. That makes a big difference. Mentorship, you need mentors. I will say that, I'm today, whatever I am today is because of the great people I got in my career or career, from starting from a school. 1 or 2 persons that change your life. See, I was never that much confident that I should go out for PhD, but my supervisor, he was more confident about me than I myself, So he forced almost, you must go, you should do this, you should do that. So you choose the institutes, where you have the environment. Environment play a great role, in your developing your career.
Yes, sir. It's inspiring, really. Yes, sir. My next question is, sir. Sir, could you please share with us some of your tips or takeaways in development of a good research manuscript, which can be understood as well as replicated by others?
Yeah. Well, there are 2 aspects to it. I'll say that for students, it's very important that, they publish it. Whatever it is, I mean, whatever the work they do, they must publish, because that's the foundation for them. Very important, masters, whatever you do, publish immediately. In PhD, whatever you do, you publish that. For researchers, you need a different, stick. For I'm not a person, who is interested in publishing too many papers. I would like to publish only, those things which make big impact. There must be something, in those papers, not for the sake of, publications. For early career, of course, they are important. I will not say that you shouldn't publish. For early career, for students and the beginning, publications are very important. But once you grow, further, I will say that you should be very selective, in publications. And target good journals, not like publish anywhere, I mean, have very simple written, manuscript, but see that you made key points, are very clear there. The key messages which you want to make from those manuscripts should be very clear. And then, give your time. Don't rush in writing and submitting. Read it again and again. Take help of the colleague. Take help of the professors. In improving them. I'll give you one example. Like after my PhD, I got this thing that early that I must publish, as early as possible. So I requested my professor that, I want to delay submission my thesis, and I want to submit my paper first, so that, I can, because that time, if I come back to India, then, it will take a lot of time in communications, because you have to send by post only. You send to your supervisor, he will correct, send by post again, and it may be lost sometimes. So I requested him that I want to do that. And then, you spend time on that. My supervisor also spent time on that. My one paper in TAG was published, without any, review. I will say that it didn't come back. It was accepted in 10 or 11 days. And simple reason is that, he spent a lot of time on that. My supervisor read it 3 times very critically, to submission. So spend quality time before submission also. Get help of, others, colleagues, and, professors in reading it. And in first go, submit a very good quality manuscript, and don't be discouraged if it is, rejected by journals, because the reputed journals, there is in some journals only 10% manuscripts are accepted, some 25%. So don't be discouraged, try it again. So,
yes, sir. It's really a stimulating word from you for budding researchers, sir. Okay, sir. My next question is, sir, you are involved in multiple projects. So the collaborative projects are invisibly associated with many challenges and benefits too. So can you share with us how these collaborations have implicated in your research programs to achieve your milestones?
This is, very, very important and very critical. Collaborations, how you develop the partnerships. A basic principle of partnership is, equal respect for all. When you work with partners, like you are sitting in a CGIAR institute, you are working with national partners, you're working with farmers, you're working with, stakeholders, other industry. You have to treat them, equal, first thing. And, see, there is a valuable contribution with each one. You have to give due credit to them. So that's the basic principle of partnership. Never show that you are the leader, you are the boss. No, all we are working for the same goal. We are all contributing to the same. So this way you can expand your partnership. And partnership is the way forward for research, specifically in plant breeding. If you want to do any research, you need to have a network of partners. And the biggest strength, I will say, the program like ICRISAT had is a partnership only. Why? I mean, I couldn't do that much in the university as I could do in the ICRISAT, simply because you have a large network of partners. You have a big facilities, you are the same person, but you can do a lot more when you have a projects where you have many partners contributing to it. Yeah.
Yes, sir. It's encouraging. Sir, one of my questions is, sir, what is your vision on farm mechanization in India? Because, sir, you have developed the machine harvesting crops for reducing the cost cutting crop production. So how will you recommend it for farming system in India?
Yeah. So for, agricultural growth in India, that's the way forward. I mean, just see from beginning, how wheat cultivation has progressed, how rice cultivation has progressed. Wheat is totally now mechanized cultivation. Rice is going the same way. Now other crops, I gave example of soybean. I'm coming from soybean state, Madhya Pradesh. It was the crop. I was thinking that the farmers will leave cultivation of soybean simply because it is labor expensive. Weeding, harvesting, very challenging. Now this is a crop totally mechanized. Same way, if you don't have mechanization in these crops will lag behind. For example, these legumes, the farmers will take the other crops which are mechanized. So we have to give due credit for mechanization. It's not only the varieties, it has to be other technologies also which make possible mechanization. So in implement sites, in all aspects, mechanization is going to be key. And I have said this at many platforms that you will see in future, chickpea will be harvested just like other developed countries in India, totally by machine in the future. And we have a big advantage. People ask me question in international platform that in India, how the farmers will afford these, combined machines and all these things. You are developing varieties, they have a small holding. No worry, we have so many entrepreneurs, you have machines available, custom hiring in every village, they are sitting there. Even the small farmers, they take, those machines on rent and, they are getting harvested. So it's not a problem for countries, developing countries, So we have to. And secondly, for some of the traits like machine harvestable chickpea variety, who is stopping you from harvesting by manually? If you want to harvest manually, they are still better than the bushy type, So we have to think that way. So I think, we have to pay highest attention on our developing technologies that lead to mechanizations in agriculture.
Yes, sir. Very meaningful concept it is. Sir, among our audience, there are many students and research scholars planning to do their PhDs or higher studies outside India or in different countries, foreign countries. So they aspire or they're inspired to get a mentor like you. So what will be your criteria, or what are the kind of qualities you are looking for a student or a researcher other than the academics point of view?
I'll say that, all the students really have, potentials. Is the recognizing potentials and giving opportunity to express the potentials. So, if you decide that, you want to go and you really make that decision, you can reach there. It will take time. It will take struggles. It's not that, sometimes that easy. Sometimes it's easy for some, but for some it takes time. But you have to make that dedicated efforts. And then you need to have very good awareness of the programs in other countries. A thorough study, where you want to go. It's not like, okay, I just want to go out country, any university, any program is fine with me. It's not like that. You are going for a higher degree, you should be choosing that. So first thing is that, you study well. I mean, now for internet, you have all information available, which was very difficult earlier, to see the area of your interest and then the work of scientists doing in that area and communicate with them. Instead of directly applying for admission in the university, start making contact with them, with your publications, contact them, write them, and then you will find that there will be many scientists, they are looking for good potential students, for their projects. That's one good way of, getting scholarships and reaching there, So I think, first thing, you choose your area and see who are working on those areas, make contact with them, and then move forward. Yeah.
Thank you, sir. It's really motivating words. Yes, sir. My last question for you is, sir, what would be your word of advice to the students and the scholars present today in our webinar who are new to the field of plant science research?
Yeah, well, see, this is never underestimate this area. Many times when, like earlier when we were studying, always, okay, medicine and engineering, it's always considered this low, this thing, but it's never going to be that low. I mean, this is going to be always important, crucial. And these areas, plant breeding, and the kind of the progress we are making, the innovations we have, the genomics and the gene editing and other areas, the development we are having, very fascinating. These are the fascinating fields. So lot of opportunities for the future. And, food and food demand and all these things are going to remain forever. And innovations have to be there. The land holding is decreasing. We have to produce more on that land. We have to be innovative in plant science. We have to do, there will be a lot of innovations coming. You can see this vertical farming and all these things, are coming. So much opportunities are there. So I will say that, first see your interest and follow that interest and move forward with this enthusiasm that, this area is never going to be, a second grade area. It is going to be always, top, preferred area, plant sciences, Yeah.
Thank you, sir. Really, this session was very interesting. And here, I would like to conclude. I would like to thank you for your time, and it was a great effort to have done to share your thoughts and experience with us. Really thankful session, sir. Thank you so much.
Thank you very much. Yeah.
So we just had a truly amazing interaction with our professor. Thank you, Dr. Sujeshri, for executing the interview that most certainly was quite intriguing. Now, wasn't that a mind-blowing session we had? So thanks a lot. You guys have been a brilliant audience. Reaching the end of this, I would like to clarify the process of attaining certification for the same. A feedback and certification link has been shared in the chat box and the YouTube comment box as well. You're kindly requested to go onto the same, fill it up, register yourselves, and thereafter, within a slot of 1 week, your certificates will be shared accordingly. And if there is some error and you do not receive your certificates after a week's time, kindly visit our official website www.plantgenomia.com. Please note that the link is valid only for 2 hours. Now, I deem it a great honor and privilege to propose the vote of thanks on this memorable occasion. First and foremost, I would like to thank our respectable speaker, Professor Pooran Gaur, whose tremendous efforts left us with an enduring effect. I now thank Mr. Alamuru for taking up such a noteworthy initiative. I would also like to acknowledge the efforts put forth by the core support team members, namely Saiteja, Aman, Shrimoy, Sumant, Deepankar Roy, and core subject team, Dr. Rupali, Dr. Sujeshri, and our remarkable audience for contributing to make this event successful. Thank you, one and all.
Thank you very much. And thanks
you
to
very
all
much, sir.
participants.
Thank you very
Thank you,
much
sir.
for joining us. Hope we
Thanks
see you
a
soon in ICRISAT.
Sure. Alamuru. Yeah. Bye to all and have a good day. All.
Thank you, sir.
Yeah.
Thank you, sir.
Okay.
and